Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 156 for the podcast. Inflammatory disease involves excessive or ongoing inflammation that damages tissues and affects many different human systems, including digestion, immunity, circulation, breathing, and the nervous system. The demand for anti-inflammatory treatments is growing quickly and is tipped to top $250 billion within a decade as cases increase and new approaches to address inflammation emerge. InflaRx is a biopharmaceutical company pioneering anti-inflammatory therapeutics by applying its proprietary anti-C5a and anti-C5aR technologies to discover, develop, and commercialize highly potent and specific inhibitors of the complement activation factor C5a and its receptor C5aR. This week, I had the chance to talk to Niels Riedemann, CEO and founder of InflaRx, about his company's first-in-class approach to fighting inflammation and what the future might hold for his pipeline in a product. I hope you enjoy my discussion with Niels. Niels, welcome to Beyond Biotech.
Thanks, Dylan, for inviting me.
Niels, can you start by walking us through your career and your journey? You've been a physician, you're a scientist, you started in sort of critical care medicine. Today, you've come all the way to InflaRx. Tell us the story.
Yeah, happy to do so. In fact, I'm a board certified general surgeon and intensivist, and I discovered my passion for science when I was a postdoc at the University of Michigan, where I met the other founder, Renfeng Guo. So we both did quite extensively researching the complement space, some of it what we'll be talking today about, and got very excited about the meaning of the C5a, C5aR pathway, this signaling that has so much to do with inflammation and how inflammation causes damage in various disease settings in to our tissues and our organs. And so, while researching this for the better of 3 years together, we the idea grew that we should probably be addressing this with suitable drugs and trying to see if this helps humans, with some of them being very devastating, others very, very impacting chronic diseases and acute diseases. So I would say, as I was moving along as being a board certified surgeon and then becoming intensivist, I actually became intensivist, even though I loved surgery a lot, because I had developed this interest in the acute care space back then, particularly in the sepsis space, and knowing that the complement pathway may play a role. So long story short, at some point we realized we may help a lot of patients eventually if we develop drugs. And so I, jumped from academia to become entrepreneur, and it was a great endeavor so far. So that's the background.
Was there a particular moment or a particular aha moment where you just decided, I'm going to move in and we're going to build our own company instead of continuing with the academic research?
I don't know if there was a particular moment. I think it was always there somewhere deep inside. And Renfeng and I met, I think a couple of years after we had done a lot of pre-work. And we both found that we're probably never going to be sleeping well, never going to be happy if we don't try it. I remember we had a conversation at the central campus in Michigan after we'd been postdocs. He was a professor. I was a young professor in Germany. And we were basically saying we still have this passion in us. We still believe we need to try it. Otherwise, we'll never forgive us for not doing it. And so I think if there was any particular moment, maybe that was it. And then we just founded the company and tried to get it funded, which we all did, and started the journey. Yeah.
So tell me about the business. What's the mission of InflaRx?
So yeah, we are developing highly specific targeted drugs to the C5a, C5aR signaling pathway in the complement field. And I'm going to be telling you just a second what that is, to address life-threatening and debilitating inflammatory diseases. So the complement system is something that is in our system, in our blood, if you will, similar to the coagulation system, which you may have heard before. But when you cut yourself, the idea of the coagulation system is eventually you form a blood clot and stop the bleeding, right? The complement system has a bit of a different meaning, but it functions very similarly. When something gets in your system that is recognized as it shouldn't be there or as being foreign, in brackets, even though it may not be foreign, but if it recognizes it as being foreign, complement system makes sure that this signal is translated into a cellular response, meaning your immune cells get it, that there's something in there that shouldn't be there and start becoming active. And they have quite a few weapons, to get engaged. And why they can get rid of whatever shouldn't be in your system. They can also unleash these weapons that can cause damage to your own tissue. When you think of the term autoimmune disease, that means you're attacking something with your own immune system in your own body that can cause damage. And so this intriguing role of recognizing there's something in your system which shouldn't be there and getting active, translating it into a cellular immune response is what fascinated us, because it is so much involved in so many different inflammatory disease settings. So our mission is develop drugs for that particular pathway, being able to control it and bring benefit to patients with devastating and debilitating inflammatory diseases.
Are you the only ones working in the field there? Are you quite unique in this approach?
I would say we're definitely first in class when it comes to the ligand, to C5a. When it comes to the receptor, we're potentially best in class. There's one other comparator that has made it to an approval situation on the receptor, an oral chemical inhibitor. And we spent many, many years, because that was really the key focus of research of Renfeng and myself, this receptor for the ligand, the C5a receptor. So there we believe we have a drug that could be best in class. So I would say quite unique in terms of really leading development in this particular space, yes.
And what about the patient load? What sort of patient count are we talking about here?
Yeah, so that can be quite different, right? As you may know, with the ligand, we have a commercial availability in the US under an emergency use authorization for a very particular small patient population that gets very, very sick, in this case, certain COVID patients, similarly in Europe. But when you think about where the mechanism can go, right, it can serve rare diseases, but also very large diseases, we believe. For example, the 904, INF904, that's the name of our small oral chemical inhibitor of the receptor that is under development to address large chronic diseases. So diseases that, maybe worldwide more than 1 million people may suffer from that, right? Like certain immunodermatological indications and other typical chronic and inflammatory autoimmune type diseases. So that's what excited us, right? We may potentially help a lot of patients. So it's interesting that may cater to both rare and very particularly damaging diseases, but also some that are widely known and may address like more general needs.
Taking something to approval and then onto market, it's always a long process, but it sometimes is smooth and sometimes it's quite rough, and there are some challenges along the way. Have you faced any particular challenges so far with InflaRx?
Oh yeah, we did. We did say. I would be happy to say it was not, but no, we did. I mean, it's always a challenge, because it's always an approach where you need to respect that regulatory agencies have their guidelines and have their strict rules. At the same time, you may have a point, or you may have a situation where it's not so easy to just like address that by quoting a guideline or a rule. So we had that, for example, in the European approval process. We only had one pivotal study. Technically, the p-value was not hitting the 0.05, so it was technically negative, but on a very explainable reason. So the challenge there, for example, to give you an example, was really to convince the committee that, the likelihood that they may approve a non-working drug was relatively low versus the likelihood that they would reject a likely life-saving working drug was higher. So that was kind of our approach to say, we understand this is not your easy task. It's like, we understand your thoughts about it too, but like we try to weigh that and say, look, in the end, there is an unmet need. There's no like related larger safety issue that should like balance it towards be careful, don't use the drug. So we always like I would say it was a bit of a masterpiece of how we try to really make the points by addressing it with data, with good science, but also with good reasoning and understanding where they come from. So I would say that was quite a unique situation that not oftentimes happens. It's rare that such an exceptional circumstantial approval is given. So we learned a lot from that. And then you have a lot of learnings with your drugs while you approach different divisions at FDA or at EMA, and you may expect responses or get responses you haven't expected, and then you just have to deal with that, like every company does.
Tell me a little bit more about the science that you're mentioning there. So in particular, we start with the C5a and C5aR, these signaling pathways. Explain why they're so critical. Why are they so important?
Yeah, so I mean, if you will, I mentioned already that this is the way that your body translates a signal into a cellular response, right? Your cells will get active. And there's something very unique about C5a. So it's not really redundant like certain cytokines are. It sits upstream of the cytokine cascade. So it means it induces a lot of cytokines depending on the cell type. Could be IL-17, could be IL-8, IL-6, and others. So known cytokines are induced by, or their production is enhanced by the presence of C5a and C5aR signaling. And at the same time, there are effects on cells that are very unique to C5a. For example, how do cells start migrating from the blood into the tissue? C5aR presence is the key initiating factor without which you don't get immune cells into the inflamed tissue. Or in the inflamed tissue, certain responses of immune cells, I'm mentioning a process here of neutrophils called netosis. So they're undergoing a certain process that causes a very strong local inflammation that is uniquely fostered by C5a. So another one that we're now looking at in a new development is more linking to when the C5a was discovered some over 45 years ago. The name that researchers gave C5a was the term anaphylatoxin. And that actually indicates that they researched it and found that it can cause anaphylactic shock and death and angioedema. So because it can strongly release histamine from different cells, that is interestingly an angle that was first discovered when they discovered the role of this factor in the blood, now called C5a. But nobody has ever tested whether that is applicable to certain allergic diseases. So we're running right now a study in chronic spontaneous urticaria, which is, to my knowledge, the first time that this mechanism is addressed in a kind of an allergic skin disease in this case. So there are very unique features with that histamine release, allergic site being, I would say, old when it comes to research, but really new when it comes to trying to address this in human disease. And then the inflammatory angle, some of it, which I mentioned, like the netosis, the initiation of the moving of immune cells from the blood into the tissue, that are really known for a long time and researched extensively, both clinically and preclinically, that addresses many inflammatory diseases. So I would say that makes this target very unique. Its central position in the inflammatory response, the fact that it boosts different responses in cells and on cytokine levels. So if you ask me personally, where would you intervene in a very strong inflammatory, disastrous situation? That's where I would intervene. And that's why we are developing these drugs and are so engaged in and believe that this mechanism hopefully may help a lot of people one day.
Your approach is a selective inhibitor for the C5aR. Is that correct?
Yes, that's very correct.
Okay. And if this, I guess if this is a place where you would start, and if this is a place where you're focused, why are you the first ones there?
Yeah, so that's really a very good question. So before I go directly to that question, I want to mention one thing, why it's so important to be selective, because that's a question we oftentimes get. Why don't you take one of the well-known complement inhibitors upstream, right? There are big success stories, big companies. The first company in the world that made complement world famous was Alexion. I think they're definitely deserve being mentioned here. And they had C5 inhibitors. And we get a lot of times the question, why don't you just take that drug? Well, turns out that these drugs cannot adequately control C5a and C5aR signaling because C5a can be produced through the complement activation, but also directly through enzymatic activation. So almost every cell can make enzymes that generate C5a, even locally. So with that knowledge, if you don't have a selective inhibitor, you won't control that pathway. And there's also real-life evidence for that in the meantime, all published. So that's a very important point. So now your question was, why are you the first one? That's actually a really good question. So I don't think we are necessarily really the first one who tried, but in terms of C5a, we are definitely the first one with an antibody that succeeded. And I would say that's a bit astonishing because making antibodies these days is almost like a fee-for-service business, right? It's not that difficult, but back in the days, it was a bit more difficult. We did this by very old school hybridoma technologies, really like design this antibody and testing it, thousands of them. But I would say the unique part about it is not making an antibody. That's not the difficult part, but this molecule C5a is very three-dimensionally active. And you need to find a way to only block the free C5a without blocking and binding to C5, which a lot of antibodies naturally do. And because if you do that, you kind of inhibit the other pathway, the MEC formation, and you don't necessarily want that. So I would say the way we found antibodies was really more because we didn't screen for binding, we screened really for activity in human whole blood, and that's how we discovered a new epitope that was not known to anyone before, that we could patent. So it was more like the path to get there because of the biology of the ligand. For C5aR, the receptor, it's a bit different, actually. There's a ton of research. We've been part of the discovery team at the University of Michigan around Peter Ward, but there have been many other groups in the world, famous researchers like Jörg Köhl and others that contributed significantly to the research. And there have been a lot of attempts, even from pharmaceutical companies, to address this. And initially, the way this was addressed was cyclic peptides. They were very potent inhibitors, but all of them turned out to be quite toxic molecules in and of itself. So they all stopped for safety reasons. On the small chemical inhibitor side, I would say, as to our knowledge, there were a lot of companies that tried and failed just to make a working drug. Then there was one company that found a particular binding site inside the pocket that allows you to make a drug that can fully block this receptor. However, and that's the marketed comparator I was referring to. However, because this is inside the pocket of the receptor, you need to be relatively lipophilic, meaning you need to like the membrane, the lipid membranes more than the blood. And so it's very hard to come to concentrations in the human system that really allows you to block it. So with that knowledge around that binding site, we designed over many years of work a drug that has different pharmacokinetic features, actually gets you to very, I would say, much better plasma levels, and therefore shows a much better coverage, close to 100% inhibition in the human system. So I would say a lot of attempts, but a lot of knowledge gained before we came to this molecule, which we are particularly proud of moving that forward in development.
You mentioned a moment ago the MAC formation, the membrane attack complex formation. Can you tell me why it's so important that you don't disrupt that with your molecule?
Yes, so the membrane attack complex formation, I mentioned C5a is being cleaved off of its mother molecule C5. So the leftover much bigger part when C5a is off is called C5b. And that C5b can assemble with C6, C7, C8, and many C9 molecules. They kind of start drilling a hole in almost any membrane they think of a mechanism that starts inserting and then drilling a hole into a membrane. If you do that to a bacterium, you're going to lyse the bacterium. It will kill the bacterium. If you do it to your own cell, it will lyse your own cell and kill the whole cell. But we are protected. We humans we have inhibitors so that this mechanism, which all the time occurs in our system, doesn't attack our own cells. There's very rare disease where this is not working, but normally we're protected. So that mechanism is an important defense mechanism, particularly against many and certain encapsulated bacteria. So if you do block it, you're going to have a black label, a warning that you need to immunize, for example, for meningococcal disease. You may also have more other types of encapsulated bacterial diseases. So that's something you don't want to have, particularly not when you dose chronically or in a setting where you have a lot of bacteria around, like in an infectious disease setting. So for the types of diseases that we try to address, we really like the fact that we keep that intact and don't have to live with the fear of strongly increased infection risks.
Tell me about the diseases that you're actually targeting. What are they specifically?
So right now with the INF904 molecule, we are going after 2 diseases in the broader immunoderm space. I mentioned chronic spontaneous urticaria already for the unique angle of that being an allergic dermatological disease because of the histamine release pathway. So that is the first time that this is tested. There are working drugs in this disease, but there's still a lot of unmet medical need. And even though this is, from a pure kind of hearing about the disease, it doesn't seem to be as horrible as a disease as some of the others. However, it's a very impacting disease. These patients have horribly itching hives. They are usually strongly impacted. Some of them can't go to work anymore. So it has a big impact on life. And it's very common. So a lot of patients worldwide suffering from that. So if we could deliver a safe oral mechanism to this disease that addresses hopefully many of these patients, that would be a big medical plus in this field and probably open more work potential in the allergic space with that mechanism that we could address later. The other disease we're going after is more one of these horrible skin diseases. I would call it hidradenitis suppurativa. I'm going to refer to it as HS because it's a bit difficult to pronounce. So HS is also a pretty common disease. When it gets very chronic, these patients suffer from severe debilitating skin lesions. They have 3 lesions. They have nodules that think of them as a deep-seated inflammatory, very painful nodule that can be up to 100 in your skin, and they can move forward and ripen into abscesses. And they can become incredibly large, painful, sometimes such that patients release themselves with knives and scissors or seek a doctor to incise. And these abscesses, drain a very odorous pus. As the disease gets more chronic, they develop very deep-seated skin lesions called draining tunnels. The term is a bit irritating because it looks like, or you think of that just being a tunnel. You have to think of this as a very strong, large inflammatory lesion in the deep skin that constantly produces pus, like a chronic abscess almost. And your body develops these tunnels to release the pus. That can bring up to a liter of pus per 1 lesion per day. As I've seen a video online, it's quite. It's extremely life impacting. These patients have these lesions mostly in the groin and genital areas. Their social life is destroyed. They retracting themselves from social life because of the odorous situation, because of the pain, because of sometimes I also say it's a shameful disease. So this disease has also approved first drugs, but there's a significant unmet need. Many of the patients who respond to these drugs lose this response and progress. And as they progress, they get more and more desperate. So that's a typical autoinflammatory kind of autoimmune disease. I did mention earlier to you, Dylan, that there's a mechanism that is quite unique to C5a, for example, the term netosis. So you induce a process similar to a cell death in neutrophils that causes a very strong inflammation. That is plenty some found around these lesions, particularly around the deep-seated draining tunnels. And the C5a receptor is expressed in various cell types strongly around all of these lesions. So there's a lot more research in HS, and we're excited about this development as we've kind of pioneered the complement inhibition kind of idea in this disease. There are other diseases we're interested in, but for lack of current finances, we cannot address all of them. We're just a small biotech company. But certainly other areas that we have also made public, for example, the chronic renal disease space, the peripheral neurological space, and others that are of potential interest, maybe to tackle with future collaborations or otherwise.
C5a has also been implicated in some other really complex diseases, things like Alzheimer's, also in some cancers. I mean, that's obviously not where you're focused now, but is there scope one day for you to partner with someone or to do some work in these areas as well?
Yeah, I mean, interesting story about, Alzheimer's disease. Some 14 years ago, I had a conversation with my former boss at the University of Michigan, Peter Ward. He said, you should look into Alzheimer's disease because there was more and more research around C5aR, C5a role in Alzheimer's disease. And while this is scientifically super interesting, I would say these are clearly challenging disease spaces from a development point of view, right? We know the story in Alzheimer's, and we know how controversial it can be, but also how high the unmet medical need is. So that's certainly not something we would tackle right now. Similarly, we have done some initial work in cancer, and this is also a very challenging situation where we believe the role of complement, particularly C5a, C5aR, will one day be more and more kind of unraveled and probably also leading to approvals of drugs. That these are challenging spaces. We need to focus. We are focusing on immunoderm right now, but particularly those two very challenging spaces, the cancer space, but also the Alzheimer's space would be probably something with the right partner to do. Or if we are one day in a situation where we can just do it on our own without that impacting the entire franchise. So high interest also personally, particularly in Alzheimer. I think that's very interesting. But you also need to know your limits. Currently, we cannot go into these fields.
You've moved through the clinic. You're in clinical trials. You've got some others in preclinical. What are the lessons that you're learning from these clinical trials, and how does that feed back into the development that you're doing?
Yeah, I would say clinical trials and clinical science are just about the most complex things because there's so many influencing factors. And, I've made extensive research experience with animal models, mice, etc. But then you go to humans and you see how diverse we are. It's amazing in our responses. So the human system is just complicated. And that's one learning. But I would say a key learning is also, I think in biotech, we have to take certain risks if we want to be on the forefront of disruption and finding things. But one of the risks that we should all be aware of, and one of the key learnings from our endeavors was that, when you're looking into a new disease area, it's really paying off if you try to understand as good as you possibly can the endpoint or the primary endpoint you're looking at. Because if you do not understand the variability of the endpoint, if you do not do an appropriate powering, you may already lose right there, even if you have an active drug. That happened to us in one field before where we didn't know about the variability, but we clearly saw a lot of signals of efficacy. Clearly the drug was active, but we couldn't show it with the primary endpoint for reasons that had nothing to do with the drug in our eyes, but really with the endpoint. So when you power your study, pay extra attention to the statistical setup and particularly to the variability in your primary endpoint. It's one of the key learnings that we made, aside from a lot of other learnings.
Niels, as the person in charge here at InflaRx, how do you see yourself growing over the next couple of years? You're going to be in the clinic more. Do you see yourself partnering more? Are you looking for expansion somewhere else? Where do you see the company going in these next couple of years?
Yeah, I mean, this is always, I would say it's a great question, but it's always also a matter of which opportunities open, right? A lot of this is always data driven. So we're currently running studies in HS and CSU. If these studies are positive, that will open up opportunities. I can see us doing these studies just as much as we can do these together with a partner. So I would say at this moment in time in the markets, I would say every company is trying to assess all options, particularly including partnering options. So I wouldn't exclude that. I would say this is certainly on the table if it makes sense for the shareholders and us and the stakeholders in the company. But it's also hard to say we're planning to do one versus the other. In the end, it will be data-driven, and I do believe if the data are positive, that we will find a way to finance next steps. We certainly have conducted larger phase 2b, even phase 3 studies, so we're certainly capable of doing it. So I would say the challenges are more like, what's the right way to finance it at that point in time if you had good data? So I can't exclude one or the other. I certainly hope to see more than just the development in HS and CSU to see if what we believe makes sense for this drug, that it could be a pipeline in a drug one day, given the mechanism we talked about today, some more, that we see this come to fruition. That would be my big goal. And I would say the big goal in the company. We would love to play more part and still a part in it. And that's what we're trying to do.
What about the landscape of anti-inflammatory treatments more generally, as well as the C5a and C5aR therapies? You've got different biologics, you've got gene therapies that are playing in that space. Do you see there being just more options or more competition, or is the market going to move one way or the other?
I would predict that we will see in certain diseases disruptions with gene therapy as well. I'm a bit more skeptical on the broad use of these therapies. We also need to think about how broadly applicable a technology is. So I would say there's a lot of space and a lot of medical need. So I'm not afraid of competition. I always think that the future may even be less just like not so much just the question of competition. I think the future will also be of what is the right combination of approaches that we know in and of itself may be beneficial, right? We haven't studied enough how you would probably combine certain therapeutics. I would say at some point that will come in the future and that will open up more benefits for patients. So I'm quite convinced about that. With respect to our mechanism, we find that being very unique. So I would hope we have a positive future in like helping hopefully a lot of patients with inflammatory diseases. But again, I'm not so afraid of competition. I would say clinical science is always difficult. We've seen some drawbacks in the I&I space with failed studies in recent months, particularly. But just as much as we've seen that, I would say we're going to see some breakthroughs going forward again. So I look at that quite positively, and in the end, this will benefit patients. So and I think the additive power of some of these technologies have not really been explored enough yet.
You mentioned these breakthroughs. You're someone who came from academia originally. Are you keeping an eye on universities and on fundamental researchers? Is that where the next breakthroughs are going to come from? Or are they going to come from industry?
That's a really good question. I think they can come from industry, but they oftentimes somewhat are linked to academia, right? If you think of our own path, our passion and our original ideas and why we wanted to do what we were doing came from academia. The actual development of the drug that we would be using later, that was in-house, right? And I think that oftentimes the missing link, we will always talk about translational science, right? And how that is so difficult. I'm probably one of the people that have done the whole thing from bench to bedside, right? I think sometimes the missing link is that you have to understand that academia works very much different from science that is conducted in companies. Because in companies, you can't fool yourself, right? You have to be sure it works. You have to be sure it's fully reliable. Research in academia can be a bit more free and chaotic in a good sense, as you may discover things that you would never discover because you wouldn't do experiments like this in a more stringent environment. So there is this kind of discovery phase which needs to be more free. And then there is also something that academia sometimes misses, the need to become much more stringent in what you do in order to address it with a workable drug. Because then it's about, can you reach the right PK? What is your distribution humans? What's your therapeutic window? Like there's a lot of questions beyond the original science that determine in the end whether you will have a working drug or not. And when this kind of understanding comes together, I would say we still need academia for breakthroughs, definitely. Can there be breakthroughs in companies? Yes, on the molecule level, there are a lot. So I wouldn't give one or the other the go. I would say both are very, very important for the ecosystem.
Just a final question: the global burden of inflammatory disease is enormous, and you spoke about that at the beginning of our talk here. But are there steps that the biotech industry can take to make sure that innovations like yours and InflaRx are actually going to be able to get to patients right around the world rather than just in Europe or just in North America?
Yeah, I think this is really a challenging question and something we think about sometimes, because there are patients that could benefit from drugs that are just not available where they are. And I would say something that may be counterintuitive at first, but if we are not willing in the Western world countries where these drugs are mostly developed, like especially in the US, particularly in Europe, if we're not willing to pay for the innovation, which I see a huge problem in Europe, right? The US being the ones that are really paying for innovation. If we think if we just cut that price down, then everybody will be helped. I think that's wrong. How do you bring the drugs then to reasonable price to these areas? There we need to be much more collaborative in terms of maybe also like allowing ourselves to say, where can we give leeway to regulations that are ultra strict on our end? What about expired drug? Can you really not extend it by a few months? Is there real hard evidence that you can't do that? You may still help patients. Maybe that's not always the case, but there's so much that could be done, which there's little incentives to do it. But one thing I also see, there are still drugs or therapeutics that been developed some 40 years ago where the hurdles were very different. And they're still making money, and they can increase their prices every year by 0.5%, 0.1% up to 1%. And they've actually never even established whether they're efficacious or even safe. So we may also want to ask our healthcare systems to say, help us to get rid of those that don't work, right? That's also not, I'm not making friends with the big pharma industry. But I would say if you want to bring innovation, you need to think also where you cut costs, right? And so innovation needs to be paid for, or it's not developed. But we need to be more ambitious and also more creative in how we bring it to those that can't afford these costs.
Niels, thanks for joining us today on Beyond Biotech.
Absolutely. Well, thanks for having me, Dylan. Pleasure.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.